Hello, guys. The topic for this week is meiosis. A couple of announcements, first of all. Week 10 extra credit quiz is due this Saturday at 11:59, so this is the quiz that's based on what we spoke about last week. Now, there is a new extra credit quiz that will be due next week at 11:59, and it's based on the material that we will cover today. And additionally, there's your week 11 PPP quiz that you have to complete in order to get your PPP points. There have been some people that may have forgotten to do the quiz or just didn't do it, and those people who didn't do it last week are not going to get points as if they were not in class. So please, guys, do not forget to do this quiz. It will be available together with this presentation. Speaking of the project, the presentations were meant to be due next week at the time of class, okay? Now, given that we're not meeting anymore, it will still be due on Tuesday at 6 p.m. or 7 p.m., depending on which class you're on. And it is just going to be a transcript of what you guys were planning to do. You have not, you do not have to meet with your teammates anymore. All you had to do is work on the outline together. And now that you've already established who was gonna do what, all you have to do is write in a paper what you were going to do during the presentation. If your presentation was a game, you have to say, I was in charge of beginning the game, and I had to ask questions about this, and that to this person. I was also going to present about Meiosis, for example, and I was in charge of talking about prophase. So prophase is blah 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 blah. Okay, that's what you have to do. But it has to be done individually. This will go through Turnitin. So again, please try not to plagiarize. There will not be a project survey due anymore, because there is nothing that you could say on the survey anymore. You're not going to be working in person with each other, so there is nothing for me to grade from the survey. Alright? So we are going to compensate for these points in another ways, like the extra credit quizzes that you've been getting so far. Lastly, there's the case study assignment. This is another writing assignment that you have to do, and it's pretty easy. You will be assigned a scientific article by me, and you have to analyze it in a minimum of 300 words. It's a simple assignment, guys. Instructions with your assigned article and rubric are posted on Canvas together with this presentation. It will be due on Saturday, April 11th by 11:59. Okay, so you've got time to do this one still. Alright, let's get started with a little bit of history about meiosis. The process of meiosis was discovered in 1876 by German scientist Oskar Hertwig, who was working with sea urchin eggs. The word meiosis is derived from the word meio, which in Latin means less, and in Portuguese means half. This will make sense later, because as we move forward, you will see that all meiosis is trying to do is reduce diploidy to half of what you began with. Let's see what this means. First of all, let's compare mitosis, which we already studied, right? Mitosis begins at interphase, and we're looking at a human cell, so we're looking at a diploid cell. It goes through prophase, metaphase, anaphase, and telophase with cytokinesis. In the end, you still get two identical daughter cells. So two diploid cells because you began diploid. Now let's look at the differences in meiosis. You begin with a diploid cell, just as in here, but you do all these steps, which are similar to mitosis. As you can see, there's prophase, metaphase, anaphase, and telophase, and there's 1 and 2. Alright, so 2 divisions occur in meiosis, but look at the difference here. We go from a diploid cell to 4 haploid cells. That is a great difference, guys. That is the great difference between meiosis. Of course, we're gonna go through each of these steps as we move forward. Haploid versus diploid. This is just a review right now. We've already established what this is last week. All organisms have at least one set of chromosomes. Unpaired chromosomes would equate to one set of DNA, while paired chromosomes would consist of two sets of DNA. Ploidy is the number of sets of chromosomes in the cells of an organism. Haploid organisms have unpaired chromosomes, which consist of complete sets of DNA. So, ploidy is just referring to how many sets we have. If we talk about humans, our ploidy is diploid because we have two sets of chromosomes. Same image that we had last week, so let's see if we catch the difference. Haploid has 3 chromosomes right here. Diploid has 3 pairs of chromosomes. So this chromosome has the same genes as this chromosome, and this chromosome has the same genes as this chromosome. But they are not identical. Okay, remember, they are not completely identical. When they are identical is when you are going to have a dyad, for example. When you have 2 duplicated chromosomes, for example. That is when you have like the X looking one, when there is a centromere. These have the same genes, but they are not equal, because one set comes from the mother, one set comes from the father. A pair of chromosomes where one chromosome comes from the maternal side and the other comes from the paternal side is a homologous chromosome. They have the same types of genes in the same location. So this is a homologous pair because they have the same genes. That is what makes them homologous, but this doesn't make them identical to each other. Okay? They are the same in what genes are there, but they can be different in what alleles of those respective genes they have. If you remember what an allele is a different presentation of the same gene. For example, if it's the gene for eye color again, there could be an allele for brown, or an allele for blue, an allele for hazel, okay? So this is what we're looking at. Each chromosome has those alleles. Duplicated chromosomes. When DNA is replicated, a chromosome makes an exact duplicate of itself. The 2 identical copies of the chromosome are connected at the centromere and known as chromatids. So now we're talking about identical chromosomes, okay? You have an unduplicated chromosome, and now you have a duplicated chromosome attached at the centromere. Meiosis is a process of cell division in sexually reproducing organisms that unpairs chromosomes in reproductive cells. Note, a cell needs to be at least diploid to undergo meiosis. This is the minimum requirement in order for meiosis to occur. Because, as we stated at the beginning, in the process of meiosis, at least in humans, and we already know that we are diploids, goes from a diploid cell to 4 haploid cells. You could not go from a haploid cell to like a 1/2 ploid 4 cells, okay? That would not happen. So it needs to be at least diploid so that you can go to something less than that. Meiosis consists of 2 divisions. The end result is 4 genetically different haploid daughter cells with half the number of chromosomes of the original parent cell. This is so important, guys. In just a little bit, it's going to make sense why we need to have haploid cells from meiosis. The goals of this is to produce gametes in animals and spores in plants. So gametes in us are a sperm for males and ova for females. Separate paired chromosomes is another goal of meiosis, and then create genetic variation among individuals. Probably the single most important one of the goals of meiosis is create genetic variation, because here, meiosis is the literal reason of why if you have a sibling, you may not look exactly the same as them. That's the reason. That is the reason why, even though your parents were the same when they had your brother and then they were the same when they had you, meiosis is the reason why you may look different than your brother or your sister. Okay? It's that genetic variation that occurs, even though you're both coming from the same two parents. Meiosis is for sexual reproduction, absolutely, always. In a diploid organism, one set of chromosomes comes from the mother and the other comes from the father. Gametes need to be haploid because when they fuse during sexual reproduction, the diploid state is restored. Now, that's your explanation, guys. So, in a woman, the cells that undergo meiosis are in the ovaries. Those ovaries undergo meiosis to produce an egg, which is haploid with only 23 chromosomes. Remember that diploid number for humans is 46, so the haploid number would be 23 in an egg. A male undergoes meiosis at the testes, where sperm is produced. The Leydig cells of the males are the ones that are diploid and then engage in meiosis to produce 4 distinct, different sperms. Okay, and they are haploid, so they have 23 chromosomes. Now, in fertilization, 23 chromosomes from the egg and 23 chromosomes from the sperm combine to bring it back to a diploid state of 46 chromosomes in 23 pairs. The embryo then continues growing through mitosis. This arrow represents mitosis here. Mitosis gives you the same over and over and over again. So we go from a diploid cell to many thousands, millions, billions of diploid cells, okay, to make the whole organism up. So all the other cells in that organism are still 46 chromosomes in 23 pairs as the zygote was. The human karyotype. A karyotype is a map of the chromosomes in a cell. Humans have 23 pairs in each cell. 22 pairs of autosomes and 1 pair of sex-determining chromosomes. Again, the sex-determining chromosomes. Aside from a couple other genes that they may have in them, most of their function is to determine whether the individual is going to be a male or a female. It's X representing female and it's Y representing male. Chromosomes pair up based on size, centromere position, and identical genes on each chromosome. Here you see a total of 23 homologous pairs, or diploid, and 46 chromosomes, which is still the diploid number. So the haploid number in humans must be 23. What are the 4 general phase names of mitosis and meiosis? Here you go. It's your regular PMAPs, okay, as we learned it last week for mitosis. Prophase, metaphase, anaphase, and telophase. A little bit of an overview. Let's not forget about interphase, okay, which is what comes before cellular division. So interphase is the first thing that we're going to look at. Meiosis 1 will be the separation of homologous chromosomes. That is the goal of meiosis 1. That is what happens at the end of meiosis 1. Then there is interkinesis. It is just the period in between meiosis 1 and meiosis 2. And then meiosis 2 is the separation of the chromatids, followed by the 4 same steps: prophase, metaphase, anaphase, and telophase. So interphase, again, we already know what interphase is, but let's have a look at it. Not a stage of meiosis or mitosis, but comes before each process. There are 3 phases in interphase: the G1, where the cell is growing; S phase is DNA replication; and G2 is necessary for proteins to be synthesized. That is where the necessary proteins for division are going to be synthesized. Everything that you need for cellular division is going to be created during G2. Now, in addition to this, there is G0, which we discussed last week, which is just cell cycle arrest, alright? Which is when the cell is no longer going to divide anymore. So it just stays stuck. Checkpoints between each of these phases ensure that everything goes smoothly and that there are no errors. Now, let's get started with the real process of meiosis. The first step is going to be prophase 1. In prophase 1, the chromatin begins to condense into chromosomes. Remember, chromatin is the same thing that makes up the chromosomes, but only more loose than this. They begin to condense, they begin to get more tight in a way that we can actually even see it in the microscope. Homologous chromosomes pair up, which this process is called synapsis, to form a tetrad. Okay, now this is already different from prophase. Alright, we're gonna try to keep track of everything that's going on in here. Let's begin here. Let's consider a human, for example. A human begins with 46 chromosomes, right? After the S phase, you still have 46 chromosomes, but now they are 46 duplicated chromosomes, alright? So now you have 46 duplicated chromosomes. What happens then is that these duplicated chromosomes can be referred to as dyads as well. One unduplicated chromosome is a monad. One duplicated chromosome is a dyad. And then when you put two dyads together, you can form what we call a tetrad. So these 46 duplicated chromosomes are going to become tetrads, which means that we're going to have 23 tetrads, okay? 23 tetrads. A spindle apparatus forms. Breakdown of the nuclear envelope occurs, same as mitosis. Microtubules attached to the kinetochores and centromeric regions, same as mitosis. And crossing over occurs at the chiasmata, not same as mitosis, okay? This does not happen in mitosis. This is where the first type of genetic variation occurs in meiosis. Crossing over is the process where genes are exchanged between homologous dyads. Let's see what this means. First, let's look at this process here. We see the chromatin condensing into chromosomes. Now they are becoming tetrads, specifically as you can see there. And that's that. Now let's dive into crossing over. Crossing over is when the homologous pair of chromosomes becomes a tetrad and they exchange genes. They quite literally take branches of each other, alright? And they mix up their genes. This occurs in prophase 1 of meiosis, an exchange of genetic information between a homologous pair of chromosomes, not chromatids. That is important because chromatids are already identical. So there's nothing to exchange. Even if they did that, then there's no end result because they are literally the same. The points at which the exchanges occur are called chiasmata. Alright, so each of these is a chiasmata now, because that is where the exchange occurred. Chiasmata. Why does this happen? To increase genetic variation. So from here on, starting from prophase 1, which is the step 1, everything after there is no longer the same as it was before. We already have genetic variation. It's already not mitosis. This is what's happening, as you can see in this video. You have a tetrad, and they exchange material. That's it. One more time. That's all. Now, metaphase 1 is the second step. In metaphase 1, the 23 tetrads that we had, 23 tetrads, become arranged on the metaphase plate. which again is an imaginary line, if you remember. It's like the equator. Each chromosome of a tetrad is attached to a microtubule from opposite centrosomes. Independent assortment is the random alignment of chromosomes on the metaphase plate that causes many different possible combinations. Now, this is very important as well. Unlike in meiosis, now each tetrad has different phases. Remember that crossing over already occurred. So independent assortment is the way in which they line up. This is better at explaining it visually. So right now, before I play it, just notice that we already have tetrads formed, and we also are able to see some chiasmata. So crossing over has already happened. What we're about to see is metaphase 1, where they begin aligning at the metaphase plate. As you can see, lining up is occurring, and they are in different positions. For example, we have purple to the left in both of them, or they could assort in any of these different ways that you just saw there. Alright? And how do they assort? It's random, guys. They just do it because they have to do it in a way, and after they are done doing it, the next step comes along, and you will see how it matters what way they were facing, because that determines which cells get which different genes. Let's see what we mean. Meiosis 1, anaphase 1 is the next step. In anaphase 1, homologous chromosomes are pulled apart toward opposite poles of the spindle apparatus. So, 23 tetrads are now being pulled into 23 dyads once again. Okay? We are making tetrads into dyads. So at this point, before the cell even separates, we had 23 tetrads lined up here at the beginning, and now we have 23 dyads going that way, and we have 23 dyads going this way. But in total, we still have the 46 chromosomes that we began with. Chromatids remain attached at the centromere and move as a single unit towards a pole. Homologous pairs officially separated, and each side of the cell now has a mixture of maternal and paternal chromosomes. This is what's happening here. The 23 dyads that we began with are being separated into the 23 tetrads that we had at the beginning are being separated into 46 dyads, each one going into a different cell. Telophase 1 and cytokinesis is the last step of meiosis 1. Each half of the cell has a complete set of chromosomes. Nuclear envelope may reform, and chromosomes may relax to chromatin. They may. They don't have to, but they may. Cytokinesis creates two daughter cells, and then interkinesis is the stage between meiosis 1 and meiosis 2. Interkinesis shares similar steps to interphase, except that there is no DNA replication occurring, no S phase. So they are not the same processes. By far, they are not the same processes, guys. Because, think about it. Now that we have, let's use this image to make it better. At this image, when they are already separating, this cell is going to have 23 dyads, and this one is going to have 23 dyads. So at this point, these cells are already haploid. They are not diploid anymore. They have 23 chromosomes, which is unlike the diploid number for humans, which is 46. So each cell has 23 dyads, so they are haploid. Now think about it. If they duplicated their chromosomes again, then wouldn't we just go back to 46 chromosomes? And we do not want that, okay? We don't want that to happen. So they stay like that. So this is what's happening there. Again, remember, we have 23 here and 23 here. So they are already haploid. So we went from 46 dyads to 23 dyads in each. So we went from diploid to haploid. In meiosis 2, the two cells produced from meiosis 1 are now haploid and will retain their ploidy through meiosis 2. So, meiosis 2, you will see that it's very much like mitosis. Technically, the same as mitosis. Alright? So it's almost as saying that the two cells that we got from meiosis 1 are now going to undergo mitosis. It's pretty much the same thing. Meiosis 2 is similar to mitosis in that you end up with the same number of chromosomes with which you started. So if we were already haploid, we're going to end up haploid. This allows for direct separation of the sister chromatids and the production of 4 haploid daughter cells, which we call gametes. That is where we get our gametes from. Males, that is where sperm comes from. Females, that is where ova come from. An independent division of each of the 2 cells produced in meiosis 1, which is why 4 unique daughter cells are produced. There you have it. That is meiosis 2. It's way simpler than meiosis 1. And you'll see why. It's mitosis. It's what we did last week. So we begin with prophase 1. Nuclear envelope disappears. They attach to the microtubules. And then metaphase 1 begins, where they begin lining up at the metaphase plate. So this is going to be mitosis. Look at it. It's the same as mitosis. So you have them lined up at the metaphase plate, and then we know what's coming after. We know anaphase is coming now. And this is where they are separated. So now, if you count, we had 23 dyads in each. Now we're separating dyads into monads. We're making 46 monads right before they pull apart, okay? Right before they pull apart, we have 46 monads. And when they are completely pulled apart, each cell has only 23 monads. Really the haploid number of humans, okay? We were already haploid because we had 23 chromosomes. Remember, a dyad and a monad are both chromosomes. So all along we've had 23 chromosomes anyway. Either monads or dyads, but 23. That is the haploid number. So in telophase 1, we're going to finish the separation, and chromosomes even relapse back into chromatin, and the nuclear envelope reappears. So now we have 23 monads in each, and they are all haploid chromosomes, guys. So we went from haploid to haploid. Everything is haploid. We went from 23 dyads to 23 monads. That is the difference. Just that instead of dyads, we have monads, but it's still 23 chromosomes and 23 chromosomes. So still haploid to haploid. The whole process was to go from the diploid first cell to haploid intermediate cells to 4 haploid finished cells. And please do notice how each cell is different from the others. Okay? In meiosis, 2 successive divisions of a diploid cell occur. They result in 4 haploid daughter cells, each with half of the genetic material of the original cell. Through independent assortment and crossing over, they create genetic variation between the daughter cells and parent cells. So, in a way that 1 diploid cell becomes 4 haploid cells. In mitosis, it's 1 single division from diploid to diploid, or from haploid to haploid. You do not need to, there is no ploidy requirement for mitosis. But remember that in meiosis you have got to be at least diploid. In mitosis you can be anything. It reproduces 2 daughter cells identical to the parent cell. So one haploid cell ends up with two haploid cells. One diploid cell ends up with two diploid cells. In mitosis, what you start with is what you end with. And in meiosis, you have the number of chromosomes of the original cell. You have the number of chromosomes of the original cell. So this little diagram here is just to show that they are different, okay? You see the gametes here are different colors. They are different genetically. Here, they are the same color. They are the same cell. So, if you have any questions about what we just saw today, please use the discussion board. I will be happy to answer any questions that you have, and I know that you will have questions about meiosis. This is probably one of the most complicated topics that we have this week, and I know you guys ask questions in class, so please don't hesitate to ask them on the discussion board. I will answer any questions that you have. Have a good day.